Ted.
All right. Can everybody hear me well? Cool. It's great to see everybody's face. I wish it was on the river over a beer or something, but everybody's living rooms is going to have to do for now. So yeah, today I'm going to share some work that we've been doing, give a little background on the ecology of the Colorado River, and then talk about sort of the science that led us to this bug flow experiment that's been going on for the last couple of years, and then show some of the early results from the first 2 years of bug flows. And yeah, get started here. So when that big dam here, Glen Canyon Dam, when that got put in, it really changed the ecology of the river. It created a totally new physical template. And so I'll just review a few of the ways that this physical template was changed. And with that change in the physical template, we've had these losses and additions of different species. And so one of the big changes is, Glen Canyon and Lake Powell now traps all of the sediment that used to flow downstream. And so we have a river that's about 1,000 times clearer. So this graph here on the left shows the turbidity of the pre-dam river. Dave Topping has sort of reconstructed what the water clarity would have looked like before the dam. And then this is what it is now. So we have a river that's a lot clearer, 1,000 times clearer. And because of that, right, we have things like filamentous algae just proliferating, especially in the upper reaches. Another big change in the physical template is the water temperature. We now have cold and relatively constant water temperatures. So that's what this graph shows here. You can tell sort of when Glen Canyon Dam was closed, right here in 63. It took a while for the reservoir to fill, but as of mid 70s, we entered this regime of mostly cool and constant water. We've had some warmer temperatures lately as the reservoir has gotten lower, but in general, we have a much cooler river. And because of that, we have things like introduced rainbow trout that are thriving in certain places. And then another obviously really big change is, the dam has regulated the flow of the river. And so gone are the big snowmelt floods that we used to have. This graph actually shows flows in 2008. And that yellow line is the estimated flow. What flow through Grand Canyon would have looked like had there been no dam. And then this turquoise line shows the actual flows. And this banding that you see is that hydropeaking or that load following. And so, we've eliminated the high and the low flows, and we have now this 3-foot tide because of hydropeaking. And these sorts of changes have allowed other things like New Zealand mud snails to thrive. We don't have good records of the species that were present in Grand Canyon prior to the dam. But a guy named Mark Vincent has sort of reconstructed what happened to invertebrate assemblages downstream of Flaming Gorge Dam. He went into different museum collections and was able to determine that at Flaming Gorge there used to be 30 different species of mayflies. And then after the dam was put in, 28 of those were eliminated. And so we don't have this same kind of record available for Grand Canyon. But this gives you an indication of some of the losses that can happen, the species losses that can happen when you completely change the physical template of a river like we have. And so that leaves us with what we call a Frankenfood web, where we have a mishmash of native species like humpback chub that are still there. But then a lot of introduced species or, just changes, things like, a food web that's based heavily on algae now, whereas it used to probably be based on leaf litter and detritus and things like that, or terrestrial material. So we have this Frankenfood web, and when I first got to GCMRC, we were interested in just trying to understand how the food web functions and how it operates. There's no analog for this food web anywhere really. It's novel, because of this new physical template and the species additions and losses. And so, working with a team of collaborators, we did these food web studies for a number of years. And we were able to do this study before and then after the 2008 flood that happened. That was the last springtime HFE that we had. And what we found was these food webs had low resistance to the controlled flood, so they changed pretty dramatically. And we think that was in part because they were simplified. Especially the invertebrate assemblage was very simplified, and so that made the web inherently unstable. And then we also demonstrated with these food web studies that fish populations appeared to be limited actually by the amount of invertebrate prey that was out there available. So there did not appear to be enough sort of bug meat to support the fish populations, or the fish populations appeared to be consuming virtually all of the bug meat that was produced on an annual time scale. So from these food web studies, well, we started going back and asking questions about, what's happened to the mayflies and the stoneflies and the caddisflies. And, this work really got the attention of the Fish and Wildlife Service, those food web studies and the conclusions from there. And so, like in a recent 5-year review on the status of humpback chub, the Fish and Wildlife Service identified the lack of an adequate and reliable food supply as the single greatest threat to humpback chub in Grand Canyon. They ranked it higher than threats of non-native predators. So some people think it does matter to have very few insects like we have. And then here's a diagram just showing sort of how impaired the insect assemblage is. We've completely lost the mayflies, stoneflies, and caddisflies really, and these are considered the canary in the coal mine in rivers in general. They're the foundation of river and riparian food webs. And so what this diagram shows is it's how many unique genera of invertebrates are present in different tailwater rivers. So this is what Fontenelle Dam looks like. There's over 50 unique genera of aquatic invertebrates, including many types of mayflies, stoneflies, and caddisflies. And then downstream of Flaming Gorge, they've lost a lot of the mayflies, but there are numerous other aquatic insect species, caddisflies and stoneflies and things like that. And then there are a few mayflies. And then, here we are in Glen Canyon. We have 12 invertebrate genera, no mayflies, no stoneflies, no caddisflies. And so, yeah, since we wrapped up those food web studies, we've been really trying to understand why are these bioindicator species, mayflies, stoneflies, and caddisflies absent. And, people have been asking this question in downstream of Glen Canyon Dam for a number of years. And really, we've been focused on this larval life stage. So studying, aquatic insects by studying the larvae. And basically what we started doing, starting in 2012 was studying some of these other life stages, things like the adults, things like eggs. And basically what we found was that when we started looking at the full life cycle of an aquatic insect, that we realized that it was actually this egg stage here ends up being incompatible with, right, these load following or hydro peaking operations. That's what's shown on the right here is a typical daily, right, flow regime where you have flows low at night because power demand is low, and then the flows come up every day. And we think that that tide is actually killing these insect eggs that are laid along the margins. And we think that's why EPT, the mayflies, stoneflies, and caddisflies are sort of absent. And so, yeah, here's a little conceptual diagram that we put together that sort of shows the big picture. We have this artificial intertidal zone now because of Glen Canyon Dam operations, where we have about a 3-foot tide. And because of that, these critical egg laying sites along the margins are gone. And because of that, we have a much simplified invertebrate prey base. And so, yeah, basically wildlife populations we think are limited by, the lack of invertebrate diversity essentially and invertebrate production. And so what we're trying to do with this bug flow experiment is push this picture to where it looks more like that on the left, right, where we have these egg laying sites are being restored, or we're providing good egg laying conditions, and then the hope is we would have more diversity of insects, and then healthier wildlife populations. I'm showing rainbow trout here, but we think this would benefit humpback chub as well, native fish, and then certainly terrestrial wildlife, things like birds and bats really rely on these adult aquatic insects when they emerge. So we're hoping with this bug flow experiment that we can make this picture look more like that on the left. And I guess I want to, yeah, just give a shout out to all the river guides and everyone that's helped us with this work. Citizen science was absolutely key to sort of painting that picture that I showed in the previous diagram. This here on the left is a picture of these light traps that folks have been helping us put out. These are set out for an hour each night in camp after sunset. And the contents are poured into a bottle and then brought back to the USGS for processing. And this diagram on the right shows sort of the coverage that we're able to get with the help of citizen scientists. So these little dark circles represent the sampling effort from those food web studies that we did. We were able to do 4 trips a year and sample at 6 different locations. These open circles, though, represent the coverage that we're getting with the help of citizen scientists. This is over a couple of years, but you can see we're able to collect samples from right throughout the canyon and almost every day during the growing season and paint a much more complete picture of what's going on. So big thanks to everyone, and I hope we can all get back on the river sometime soon and get back to collecting cool data. So yeah, that led us to this bug flow experiment. So we showed that load following operations were creating this bottleneck for insects. It was killing the egg stage, and we think that was why diversity and production of insects was low. And so we wanted to try and translate that into into a policy, into some change, a way to mitigate that undesirable effect. And so what we came up with was this bug flow experiment. And the purpose is essentially to improve the egg laying conditions for insects. And therefore, we're hoping to see increases in the abundance of midges, which are a species that's present and a really important food item for humpback chub and other native fish, but their production tends to be low. Over the long term, we're also hoping to see increases in the EPT diversity, the mayflies, stoneflies, and caddisflies. And then ultimately, right, we're hoping to see a healthier food base that translates into healthier wildlife populations, things like humpback chub and other birds and bats and things like that. And so this shows the design of the bug flow. Basically, On weekends, flows are held low and stable. They're close to the minimum value, you know, for yeah, during normal sort of weekday operations, right? We all know that flows tend to fluctuate between a minimum and a maximum. And so on the weekends, the flows are held steady at this low value. And the hope is that that provides good egg laying conditions for these aquatic insects every weekend. We don't think, you know, eggs that are laid on a Sunday are going to hatch by Monday, but because the flow level's at the low, those eggs are going to stay wet and they're at least going to have a chance to hatch, right? And let's see. And then by doing this just on the weekends, it minimizes impacts to hydropower. So this experiment ends up being pretty neutral to hydropower. And so, yeah, this bug flow experiment was tested in 2018 and 2019, the months of May through August. Every weekend, the flows were held low and steady. And then I'm going to try and switch over to a video real quick that we got showing the this egg laying. So let's see, bear with me here. Okay, can y'all see this screen now that says Wednesday, May 9th? Cool. Okay, so I'm going to start this video. So we borrowed, let's see here. We borrowed a time-lapse camera from Paul Graham's and set it out during the first weekend of bug flows. And this is up in Glen Canyon. And so this is showing Sunday water at a spot up at 13 Mile. Aquatic insects love these emergent rocks for their egg laying. And so you can see a whole day of just steady water. So now this is Monday, a return to the fluctuations. And you can see this, this yellow banding here. These are tens of thousands of eggs that were laid down on Sunday, essentially. And you can see, so this is typical Monday flows. Water comes up, you know, by 10, 11 o'clock. All these rocks are just underwater. Now keep your eye on that little red circle. This is a spot where a bunch of midges went to town overnight. So you see that little patch of eggs there, right? These are eggs that were laid down on Monday, right, when the water was high. These insects tend to lay their eggs at dusk or after dark. So you can see that little patch of eggs. That was out of water for, you know, 6 hours, 8 hours. And so those eggs are totally dead. And so that sort of shows you the process, right, that we were trying to enhance with these bug flows is that egg laying process. And so, you know, like we see on the left here, during bug flows, we had a lot of egg activity right at the water's edge. Whereas during load falling, we had eggs getting laid high and dry, right? And these eggs can't handle anything more than like an hour of exposure to air. So, and then, you know, the other thing we hadn't sort of predicted or anticipated was just that there's a lot more egg laying habitat, right? During those bug flows, those emergent rocks are out there and ready. Let's see, screen sharing has stopped. Okay, sorry about that. So I'll go back to the presentation now. Is everybody with me still? Cool. So yeah, so here we are. Let's see. And you guys are seeing the graph now? Cool. So, so yeah, insect response to bug flow. So this flow experiment's happened for 2 years now. And we had a 6-year baseline, right, of sort of pre-bug flow light trap catches. And we, you know, midges were generally the dominant species that we were catching in these light traps. And then here's these caddisflies. This is one of these EPT species that we were really hoping would benefit from this flow experiment. And so, you know, first year of bug flows, what did we see? We saw a 400% increase in these caddisflies, right? And that was just amazing. And then, you know, the next year we're thinking things are going to be just through the roof. And, you know, what happened actually was we just came back down to earth in 2019 on those caddisfly numbers. The midge numbers were up a bit, which was encouraging. But this really got us scratching our heads, right? Like, how can we go 400% increase, you know, exactly the kind of thing we were hoping for, and then just come way back down to earth? And so one of the things we started looking at in terms of diagnostics, you know, trying to understand what happened is we looked at the phenology, which is the study of the timing, the seasonal timing of things like insect emergence or when plants bloom. And so we started looking at whether there was differences in the timing of when these adult caddisflies were emerging from the river. And what we found was, yeah, there was huge differences actually. So in 2018, the peak caddisfly emergence, that's what's shown here, is sort of the catch rates of caddisflies in those light traps across different months. The peak happened in early July in 2018, whereas in 2019, we didn't get the peak until August 13th. So there was a 1.5 month difference in the timing of peak emergence. For midges, it was even more dramatic. It was a 3.5 month difference between emergence. And emergence timing is an indicator of the growing conditions. And so that suggests that the growing conditions in 2019 might have actually been really poor, this late emergence. And so, you know, I guess to try and help sort of make sense of all this, we've got this conceptual model, you know, that we think about. Right, here's our invertebrate prey base, and we know that there's lots of things that can affect that prey base, things like the amount of algae that's out there, other things like that. And what we're trying to do with this bug flow, right, is directly affect these insects using flow. But the question is, like, what happened to these other environmental drivers in those years in 2019? Maybe those were much different, you know, across the years of this bug flow experiment. And so one, you know, environmental driver that we might expect to determine the timing of emergence is water temperature. And so we're comparing here the timing of emergence that's shown on this y-axis here. Each one of these points represents a different year in our data set. And it shows no relation to water temperature for either midges or caddisflies. So it doesn't appear that differences in water temperature across 2018 or 2019 can explain what happened. But if we look at the mud, how muddy was it over the spring? That's what's shown on this graph. So this x-axis represents how much mud cumulatively was in the river during the spring down at National. And this is a logarithmic scale, right? So in 2018, it was super clear conditions. We saw really early emergence. In 2019, over here, we had 10 times more mud in the river. And as a consequence, we had a much, much later emergence for both caddisflies and midges. And so we think that, you know, the reason we came back down to earth in 2019 was the growing conditions in the spring were just really terrible for these caddisflies and for midges. And so, let's see. So yeah, so then, yeah, basically, you know, the Bureau of Reclamation and the Adaptive Management Program, they agreed to do a third year of this bug flow testing. We had sort of one really good year and one mediocre year. And so this year, 2020, is thought to be sort of the tiebreaker. And so we've been having this bug flow experiment since May. And unfortunately, though, we're not able to do any monitoring, as you know, because there's no river trips. And so we're going to have a bit of a blank spot in our citizen science data set. But we think we'll be all right. If we can get back on the river and collect a bunch of data starting in mid-June, the hope is we could still catch the caddisfly peak and be able to describe what happens. And so we're crossing fingers that we get some good data eventually. And so, yeah, conclusions, I guess life history, it really informs species management. I think sometimes, biologists tend to focus on a certain life stage of an animal. And, I guess one lesson from this talk is that, we got to think about the whole life cycle of these animals. And then another, take home is win-win solutions appear to still be possible. This bug flow approach is super low cost for hydropower, and we're hoping it leads to benefits to aquatic insects too. And then finally, citizen science. That's a really powerful and low cost tool for monitoring. And so that's all I got. Happy to take questions if there's any time.
Okay, well, why don't you stop your screen sharing and we can.
You got it.
Yeah.
Thank you, Ted. That was great.
Hey, glad you liked it.
So if you guys have any questions,